You've likely taken a course of antibiotics before at some point, right?
Absolutely. Most of us have.
I mean, you go to the doctor with a terrible sinus infection or maybe a UTI, and they just hand you a prescription.
Right, old piece of paper or a digital script.
Exactly. And it says to take, say, 500 milligrams of this specific pill
Hmm.
twice a day for a week. And, we just accept that. But if you stop and really think about it,
How did they actually arrive at that number?
yes. How exactly did your doctor know how much of that drug it would take to actually stop your specific infection? I mean, why 100 milligrams? Why not 50 or like 5,000?
It's a question we completely take for granted. We assume there's this universal biological rule book sitting on a shelf somewhere.
Right, like a master spreadsheet.
Exactly. But the reality of how that specific dose is calculated for your specific infection is far more dynamic. And honestly, it's far more fragile than most people realize.
Well, okay, let's unpack this, because for today's deep dive, we are looking at a deceptively simple concept that underpins literally all of modern medicine. And that is the minimum inhibitory concentration, or MIC.
The MIC, yes, it's the cornerstone of it all.
We've got this fascinating stack of sources today, spanning historical microbiology notes to international clinical standards and modern pharmacology. And we're going to figure out how a microscopic event in a tiny plastic well basically dictates international health policy.
It really does.
Because this isn't just dry lab trivia, This is a global, high-stakes biological chess match.
It is. And what's really fascinating here is how a localized, almost invisible event scales up. I mean, our goal today is to bridge that gap, connecting a single test tube to the massive global fight against antibiotic resistance.
Right.
But to do that, we have to understand what this actually looks like on a laboratory bench first.
So before we can understand how this whole MIC thing saves lives, we need the ground rules. What exactly is the minimum inhibitory concentration?
Formally, the MIC is the lowest concentration of an antimicrobial agent that prevents the visible growth of a microorganism.
Okay, visible growth.
Right. And this is usually expressed in micrograms per milliliter or milligrams per liter. And this prevention of visible growth has to happen after a standardized period of incubation.
So they don't just check it right away?
No, typically it's 16 to 24 hours. And it has to be at human body temperature, so roughly 35 to 37 degrees Celsius.
I'm picturing like a sort of limbo bar for bacteria, or actually maybe a ladder makes more sense.
A ladder is a great way to look at it.
Yeah, so you take a bacterium and you expose it to a ladder of doubling drug concentrations. So you have a tube with 0.5 micrograms, the next has 1, then 2, 4, 8, 16, and so on.
Doubling at each step, exactly.
Yeah, but incubate them overnight, and the next day you just look for the lowest rung on that ladder, where the liquid broth stays perfectly clear instead of turning cloudy,
Yeah,
right?
yes.
The cloudy tubes mean the bacteria, well, they threw a party and multiplied. And the clear tube means the drug actually stopped them.
It is essentially a miniature survival experiment. You are literally titrating the drug against the pathogen to find the exact tipping point.
But wait, I have to push back on something of that formal definition you gave. You said it prevents visible growth.
I did, yes.
I mean, visible sounds a bit subjective for modern, rigorous science. Are lab technicians really just holding test tubes up to the ceiling light and squinting at them?
That raises a really important point. It is a known tension in the field. That deceptively simple definition hides a lot of contested territory.
That
Because what counts as no visible growth is absolutely a judgment call sometimes, especially because of a phenomenon called trailing.
trailing, like they're leaving a physical trail behind.
Think of it more like a faint haze.
Oh,
With certain drugs, notoriously the azole antifungals and echinocandins, the organisms don't just stop growing completely at a sharp cutoff.
they just kind of slow down.
Yeah, instead they grow in this fine, subtle haze, even at inhibitory concentrations. It isn't fully cloudy like the control tube, but it isn't perfectly clear either.
Wow, that sounds like a nightmare for standardization.
It really is. Because of this subjective haze, international testing bodies have had to write incredibly detailed, strict rules about exactly how much haze is tolerable before you officially call a well positive for growth.
That is wild. So there's an entire bureaucracy out there dedicated just to defining a cloudy test tube.
Absolutely. There are manuals dedicated to it. And to make sure that test tube is standardized across the world, the core idea requires starting with a very specific number of bacteria.
Right, you can't just dump a random amount in.
Exactly. By convention, that starting number is roughly 5 times 10 to the 5th power colony forming units per milliliter.
Okay, so that translates to about, what, 500,000 bacteria?
Correct, 500,000. And that specific density was chosen very carefully. It is dense enough that you can reliably see the growth if it happens.
But dilute enough that the antibiotic isn't just physically overwhelmed by the sheer mass of the bugs.
Precisely. Now, to run this miniature survival experiment, there are 3 main practical methods you're going to see in labs today.
Okay, lay them out first.
The first one is the gold standard. It's called broth microdilution.
This is what I was picturing with the ladder, right? But miniaturized into those tiny plastic 96-well plates.
Yes. Each little well has a different concentration of the drug in a liquid broth. It is highly precise and highly quantitative.
But I'd imagine setting up 96 little wells by hand is pretty tedious.
Oh, doing it manually is incredibly labor-intensive. So routine hospital labs usually rely on commercial pre-made plastic panels.
Makes sense. What's the second method?
The second is agar dilution. Here, instead of liquid broth in tiny wells, you mix the drug directly into solid agar jelly plates at those different doubling concentrations.
So you have a plate for 1 microgram, a plate for 2, a plate for 4.
Exactly. And the advantage there is volume. You can spot test dozens of different bacterial strains on the exact same jelly plate at the same time.
Oh, I see. So it's great for mass surveillance. Like if you're testing hundreds of different bugs against one single drug to see general resistance trends.
Precisely. It's highly efficient for large surveillance studies. But as you can guess, it's totally impractical for a hospital trying to treat one single patient at a time.
Right. Which explains the absolute brilliance of the third method you see in the sources, the E-test, or gradient diffusion.
Ah, yes, the E-test.
Yeah, because instead of discrete steps like jumping from 1 to 2 to 4, this Swedish company, AB Biodisc, which is now bioMérieux, they figured out how to impregnate a plastic strip with a continuous, seamless gradient of the antibiotic.
It really is brilliant engineering.
It's so cool. You take an agar plate, spread the patient's bacteria all over, and just lay this plastic strip down.
And as the drug diffuses outward into the jelly, it creates this clear elliptical zone where the bacteria just can't grow.
And the best part is, the strip has a scale printed right on it. So you literally just read the MIC number right where the edge of the bacterial ellipse intersects the strip.
Exactly. It gives you the precision of the liquid microdilution method, but with the hands-on simplicity of just dropping a piece of plastic paper onto a plate.
Okay, so we've got the methods.
Yeah.
We have this clear liquid in a plastic well, or this numbered plastic strip. But, if I'm the patient lying in a hospital bed, how does that raw lab number actually dictate my fate? Like, how does it change my treatment?
Let's consider the everyday urinary tract infection, or UTI. It's often caused by E. coli.
Right.
Hospital labs run MIC tests for E. coli against fluoroquinolone drugs like ciprofloxacin constantly, every single day.
And resistance to those drugs is, unfortunately, rising everywhere.
It is. Now, if your lab runs the test and the MIC comes back comfortably low, it means it took very little drug to stop the bacteria.
So that isolate gets the label susceptible.
Yes. And for you, the patient, that means the doctor writes a prescription for a simple oral pill. You go home, you take it for a few days, and you are totally fine.
But what if that number is higher?
Well, if the MIC creeps up toward or past the resistant breakpoint, that simple oral pill is going to fail.
Oh, wow.
The doctor is now forced to switch to alternatives. And those alternatives might be much broader spectrum, they might have terrible toxic side effects, or they might require you to actually be into the hospital for 5E therapy.
So that single MIC number literally redirects your entire treatment plan and your life for the next week.
Exactly.
But sometimes, reading the sources, it seems like the physical reality of the drug itself makes getting that simple number incredibly difficult. Like take colistin, for example.
Ah, colistin.
Yeah, it's used against these terrifying, multi-drug resistant, gram-negative superbugs, like Klebsiella pneumonia or Acinetobacter baumannii.
Yes, the true last resort pathogens. For some of these infections, colistin is quite literally the only drug left on the shelf that works.
But colistin is this massive, bulky molecule. So because it's so physically large, if you try to use a traditional diffusion test like putting it on agar or using paper discs, it completely fails.
Right, it doesn't diffuse.
It just doesn't spread through the jelly properly. So the underlying chemistry of the drug literally breaks the normal testing toolkit.
Which is exactly why international guidance bodies had to step in and mandate that broth microdilution, the liquid method in the tiny wells, is the absolute only acceptable way to test colistin.
You just can't use agar at all.
No, you
Yeah.
cannot. It's a perfect example of how the physical properties of the medicine force the laboratory's hand.
Okay, so the lab gets the number, however they have to get it, and then they assign a label to it. Susceptible, intermediate, or resistant. But those labels aren't just, handed down on stone tablets. The story of MIC creep with vancomycin really highlights this.
That is a classic case. Vancomycin and Staphylococcus aureus in the 1990s and 2000s.
Right.
Researchers started noticing that the MIC numbers for vancomycin were slowly drifting higher over the years.
But, and this is the crazy part, they were technically still sitting inside the susceptible category, right? Like they were at maybe 1.5 or 2 micrograms per milliliter.
Yes, technically still susceptible according to the charts.
So what does this all mean? If the bacteria are still officially labeled susceptible by the rulebook, why does a slightly higher number even matter?
That is exactly the dilemma the medical community had to grapple with. Because if the rule book says 2 is susceptible, the doctor assumes the drug will work perfectly fine.
Naturally.
But when they looked at the actual patients, they found that people infected with these higher but still officially susceptible strains were having significantly worse clinical outcomes.
So the vancomycin was failing in the real world.
It was. The label was completely hiding the reality.
Wow.
The susceptible and resistant label alone was masking clinically vital information. The bacteria were slowly adapting, pushing the MIC up, and the human rulebook just hadn't caught up yet.
So what happened?
This real-world failure eventually forced standard setters to physically rewrite the breakpoint tables. They actually had to lower the cutoff for vancomycin.
They literally moved the goalposts because the bacteria were winning.
They did!
And that perfectly highlights that the label attached to the MIC is just a human invention. Which I think leads us directly into the fierce debate over who actually gets to set those labels.
Oh, yes. You are talking about the Great Transatlantic Divide.
The big fault line in microbiology.
Exactly. The split between the major standardizing organizations. On one side, you have the CLSI, the Clinical and Laboratory Standards Institute in the United States. And on the other, you have ECAST, the European Committee on Antimicrobial Susceptibility Testing.
These are two massive organizations,
Yeah.
and they do not always agree on where to draw the line.
No, they do not.
You can have the exact same bug tested against the exact same drug, producing the exact same MIC number, let's say it's 2. In an American hospital following CLSI, that computer spits out susceptible, and the doctor gives the drug.
But if you take that exact same Petri dish on a flight to Paris, where they follow UCAS breakpoints, the computer reads that same 2 and spits out resistant.
And the doctor withholds the drug.
Exactly.
It honestly blows my mind that a purely physical measurement is so subject to bureaucratic interpretation. I mean, a country using stricter rules will look like it has a massive antibiotic resistance crisis, even if their actual bacteria are identical to a neighboring country.
It's true. And if we connect this to the bigger picture, it really reveals how much of medicine is based on expert consensus rather than absolute physical laws.
Right.
And that gap between the raw physical number and the human interpretation of it leads to a lot of really dangerous misconceptions.
Well, let's talk about those, because if the rules literally change when you cross the Atlantic Ocean, it makes me realize how many assumptions we make about these drugs.
A lot of assumptions.
For instance, let's do some rapid-fire myth-busting based on the sources. Misconception 1. I always just assumed a lower MIC meant a more powerful drug.
A very common thought.
Like, if drug A has an MIC of 0.5 and drug B has an MIC of 8, drug A is the ultimate killer.
Absolutely false.
Okay, why?
You cannot compare MIC values across different drug classes because drugs behave very differently in the human body. A drug with an MIC of 8 is actually vastly superior to one with an MIC of 0.5.
Wait, really?
Yes, if you can safely give the patient enough of that first drug to reach a blood concentration of, say, 100. Meanwhile, that second drug might become fatally toxic to the patient if it goes above a concentration of 1.
Oh, so what matters isn't the raw MIC number at all. It's the MIC relative to how much drug you can actually get into the patient safely.
Exactly.
Okay, that makes a lot of sense. Misconception 2. I want to go back to something you said right at the start. You defined MIC as preventing visible growth.
I did.
Does that mean the drug isn't actually killing the bug?
That is another crucial misconception. MIC only measures the inhibition of visible growth. It does not necessarily mean the bacteria are dead.
So they're just chilling.
They're paused, they can't multiply, but they aren't dead. There's actually an entirely separate metric called the MBC.
The minimum bactericidal concentration.
Right. The MBC is used when you need to measure actual killing power, which formally requires a 99.9% reduction in the bacterial population.
So with MIC, the drug just hits the pause button, and our own immune system acts as the cleanup crew.
In many, many cases, yes. Drugs that just pause growth are called bacteriostatic. And for many common infections, like when we use tetracyclines, pausing them is totally enough. Medicine is really a partnership between the drug and the host's immune system.
That completely changes how I think about taking antibiotics.
Right.
But okay, misconception 3. Surely, if the lab report says susceptible, the patient is guaranteed to be cured.
Right.
I mean, the test worked.
I really wish it were that simple, but remember, the MIC test happens in a nice, clear liquid broth.
Right.
It doesn't account for the blood-brain barrier. It doesn't account for a bacterial biofilm clinging to a prosthetic hip joint.
Or poor blood circulation, like to a diabetic ulcer.
Exactly. Susceptible is just a probability. It's based on average drug levels in average patients.
Yeah.
It is absolutely not a promise.
Okay, so if susceptible isn't a guarantee, what else is this simple overnight test missing? Because clearly, it's missing a lot.
The first major missing piece is the dimension of time. And this was really championed by pharmacologist William A. Craig in the 1980s and 90s.
Ah, Craig's work.
Yes. He proved that simply knowing the MIC is necessary, but totally insufficient on its own. You need to understand the drug's pharmacokinetics and pharmacodynamics.
The PK/PD indices.
Exactly. PK/PD.
Basically the dosing science,
Yeah.
like how the body affects the drug and how the drug affects the bug.
Spot on. You need to know how the drug's concentration behaves over time inside the body. For example, for a class of drugs called aminoglycosides, you need a massive peak.
A peak.
You want the maximum concentration to absolutely dwarf the MIC, but just for a short moment. It's like a sledgehammer blow.
And that's known as the Cmax to MIC ratio, right?
Yeah.
The maximum concentration compared to the MIC.
Yes. But if you look at beta-lactam antibiotics, like penicillin, a single massive sledgehammer blow doesn't work at all.
Really?
No. They depend entirely on time. You need the drug concentration to stay moderately above the MIC for a long, continuous percentage of the dosing interval.
So it's less of a sledgehammer and more of a steady vice grip.
Exactly. Time above MIC. If you use the sledgehammer technique when you actually need the vice grip, the MIC doesn't matter at all. The treatment will fail.
Wow. Okay, here's another wild exception I found in the notes, and honestly, this one is terrifying. Heteroresistance.
Yes. The lurking subpopulations.
So a standard MIC test is measuring the bulk behavior of that 500,000 bacteria in the tube.
Right, the hour's response.
So if 99.9% of them are susceptible, the tube stays clear, the lab says susceptible, and everyone high-fives. But what the standard test entirely misses is the physical reality that maybe a tiny fraction, like literally a few dozen bacteria in that tube, are fiercely resistant.
It is a notorious problem, particularly with colistin, which we discussed earlier.
Just sneaky.
Think of it as an evolutionary ambush. You give the patient the drug, you wipe out the 99.9% of normal susceptible bacteria, and suddenly that tiny lurking subpopulation has zero competition.
They have all the nutrients, all the space.
Exactly. So they explode in numbers, they completely take over, and the patient crashes. And the standard lab test gave you absolutely zero warning because those few dozen bacteria weren't enough to make the tube visibly cloudy on day one.
That is deeply unsettling.
It keeps microbiologists up at night.
Well, okay, here's where it gets really interesting, and this is my favorite part of the deep dive. The Eagle effect.
Oh, Harry Eagle. Yes.
Named after Harry Eagle from his work in the 1940s and 50s.
His experiments completely upended our basic intuition about pharmacology.
Because human intuition says, if some drug is good, more drug is better, right?
Naturally.
If an MIC of 2 stops the bug, blasting them with a concentration of 200 should obliterate them instantly. But Harry Eagle found this crazy paradox. With certain beta-lactams and drugs like daptomycin against Staphylococcus aureus, if you use super high concentrations, you paradoxically kill fewer bacteria than if you used a moderate dose.
It is baffling at first glance. I mean, how does more poison equal less death?
Exactly. How does that physically happen?
Well, the mechanisms vary depending on the drug. But essentially, by blasting the bacteria with too much drug too fast, you force them into a defensive state.
You scare them into hiding?
Essentially, yes, you alter their metabolism. Beta-lactam drugs, for example, only kill bacteria that are actively growing and dividing.
No.
So if a massive, overwhelming dose of the drug instantly shuts down their cellular machinery, they stop growing. And because they stop growing, they actually protect themselves from the secondary lethal effects of the antibiotic.
So by trying to nuke them, you accidentally trigger their bunker defense system.
Precisely. And this really underscores a core belief in infectious disease. Reading dosing data simplistically is dangerous.
You have to look deeper.
Critical thinking is absolutely required. You can't just blindly throw more chemicals at a problem and expect better results.
As we zoom out from Hairy Eagle and these wild lab paradoxes, let's look at the global scale for a second.
Right.
Because this whole framework had to start somewhere.
It did. And the origin of all this really traces back to the sheer panic of the 1940s.
Right. Alexander Fleming discovers penicillin in 1928, but it isn't until World War II that it hits mass production.
Exactly. And almost immediately, within just a few years of civilian use, doctors start seeing penicillin-resistant staph aureus.
It happened that fast.
It did.
Yeah.
They desperately needed a way to figure out who to give the scarce drug to and who it would fail on.
And out of that sheer necessity, the tube dilution method is born.
Yes. And if you fast forward to today, that simple 1940s logic finding the clear tube feeds into everything.
It's everywhere.
Hospital antibiotic stewardship programs rely on aggregated MIC data, called antibiograms, to decide which drugs to stock in the pharmacy and which to restrict.
And at the highest level, the World Health Organization's GLAS system, the Global Antimicrobial Resistance and Use Surveillance System, tracks the entire global resistance crisis using this exact data. It's incredible. The invisible machinery powering international public health is just millions of tiny plastic wells and clear liquids being read overnight in hospital basements.
It really is a marvel of collective human effort and scientific standardization.
It is. So to everyone listening, the next time you look at a simple orange pill bottle or a doctor tells you to take 500 milligrams, I want you to picture that vast global infrastructure.
Keep it in mind.
Picture the serial dilutions, the transatlantic debates over breakpoints, and the massive surveillance networks humming along while you sleep, all just to find your specific, perfectly calculated dose.
It's a lot of work for one pill.
It really is.
Yeah.
But I want to leave you with one final, slightly unsettling thought to mull over.
Oh, boy.
We talked about how the entire MIC system relies on starting that lab test with exactly 5 times 10 to the 5th power bacteria, 500,000.
Right, the standard inoculant.
But what happens if you get a severe real-world infection? Like, say, a massive walled-off abscess in your tissue. In that abscess, the bacterial density isn't a neat, polite 500,000.
No, it's not.
It could be massively, exponentially higher. We are talking billions upon billions of bacteria crammed together.
This is known in the field as the inoculum effect.
Yes, the inoculum effect. And there is evidence that when the bacterial burden is that unimaginably high, the true MIC required to stop them spikes dramatically.
The drug just gets physically overwhelmed.
Exactly. Overwhelmed or even destroyed by defensive enzymes, the bacteria producing en masse. Which means your doctor could be looking at a lab report that says susceptible. A report generated using a polite 500,000 bacteria, while inside your body, the sheer volume of the infection has rendered that report completely, dangerously useless.
It's a very real blind spot.
It's a chilling reminder of the gap between a perfectly controlled petri dish and the messy, unpredictable reality of the human body. Just think about that the next time you pop an antibiotic.